and Dickinson. So I'm going to talk about lab automation, and I'll use the BD Keystra system as a model because that's the most developed of lab automation. But I would anticipate that over time we're going to see other automated platforms being introduced. And so I think as we evaluate these, what we should really be asking is not what these systems, how these systems can replace us, but rather how these systems can improve what we do. And I think that's the real value of automation, looking at being able to do things that we couldn't do before. And I'm going to just give a few examples of where that is the case. So this should not surprise you that much of what we do today is what Louis Pasteur was doing in his laboratory. In some cases, I've seen labs that look not much different than that, maybe a little bit messier, but I think we have plenty of room for doing things in a different approach. Keystra System is a Dutch company that we bought in 2012. I think they were truly gifted innovators, and by their own admission, they were not microbiologists. In some ways, that allowed them to innovate without the constraints of the way we would traditionally look at how we should be doing microbiology. I've also been reminded many times by the head of our R&D department that BD is really an engineering company that works in microbiology. So I think we can look at Keystra and say they were an innovative company that entered the microbiology field. Now Keystra was actually involved in microbiology for more than 30 years doing automation, but it was in industrial microbiology. It was in 2006 that they introduced clinical lab automation. And what I'm showing here is the current versions of Inocula, the specimen processing component, the WCA, which is the inoculating component that's tied with a track to incubating systems, and then the TLA, or Total Lab Automation, which is everything there plus tracks to stations that allow you to work up specimens. And I'll give a little bit more detail on each of these. Now, if we start with the Inocula, this is a picture of it, and it has an area where plates are stored. It has an area where specimens are introduced, a fully automated workstation, and a semi-automated workstation. The semi-automated workstation shows what looks like a biosafety cabinet. That's by design. The portion next to it, the fully automated workstation, also has filtered air, laminar flow, HEPA filtered laminar flow air in that portion. What I'm not going to talk about is the number of different types of specimens or any of that type of detail, which obviously is important, but just talk about some of the unique things. Now, we traditionally inoculate plates with swabs and loops, and we have no control over really how much specimen is put onto those plates. One of the innovations that was introduced, and maybe it's not all that innovative, it's just looking at things from a different perspective, is to dispense your specimen with pipettes, where you get very accurate dispensing of the sample that's being looked at. We also have developed a way of monitoring that the specimen actually is dispensed. You can see the sample as it's dispensed with video imaging and be able to measure that, in fact, that it was applied to the plate. So this increases the reliability of a very important component of what we do in the laboratory. And I'm sure that those that work in the laboratory here have seen plates that appeared not to have been inoculated or were inoculated with a volume that's much smaller than what was seen on other plates from the same specimen. All of that is eliminated with this type of an approach. They also decided that using loops was not necessarily the best way to streak a plate. And in this example, if you looked at the area that you can accurately streak with a loop, it's about 80 centimeters. If you use a magnetic bead to spread that inoculum, it's about 400 centimeters. And the longer that streak area that you can apply, the greater your chance of getting isolated colonies. And if you have more isolated colonies, you do fewer subcultures, and you're able to do more definitive identification and susceptibility tests because you have pure colonies that you can pick and analyze. And all of the studies that have looked at streaking with magnetic beads, keystro streaking, have demonstrated that value. The other thing that's related to this is the actual size of the material that's being applied to the plate. And with a loop, it's about 4 millimeters squared, and the area on the ball that's in contact with the auger is 78 millimeters square. So if you think about a loop, unless you take the loop and you streak it on one side, then streak it on the other side, then streak it along the sides, you're really missing a large portion of the inoculum that was put onto that loop. With a bead, it doesn't matter because it's rolling over the surface. So everything that was on that bead is applied to the surface. And again, if you think about it, it's the easiest in concept. Let's say instead of streaking back and forth on a plate, you're able to just make one long line. Because you've put with the keystro system, because you put a known volume of material at the beginning of that streak pattern, and then you streak it out in a linear way, as you get further away from the initial inoculum, you're going to have fewer and fewer bacteria or yeast that are stuck to that ball. And so by measuring the distance from the beginning to where you have isolated colonies, you can actually calculate the original inoculum that was on that ball or in your specimen. So in essence, you can do quantitative cultures in every specimen you apply with that ball. Now, that's primarily important for things like urines and BALs, where you want to be able to quantitate how many bacteria are there because we have criteria for how we work up specimens. But it may also at some time become important for other types of specimens that we do semi-quantitative estimates now. But it just shows the potential for using this type of a streaking pattern for doing things very differently. Now let me shift gears a little bit, and we'll talk about then what a WCA or a TLA, work cell automation or total lab automation, can allow you to do. And I'm really going to focus now on the incubator, because I think the incubator becomes, in my mind, the most important thing, most important component of lab automation. Now, these automated incubators allow you to take a plate into the incubator, to put it on a specific shelf, which allows then air to circulate around the top and the bottom of the plate, which is not what happens when you stack plates in your incubator today. What that allows you to do is get better air circulation, and because the plates never leave that incubator until you want to do something with them, like identify an organism or do susceptibility test, and because the door never opens, you have uniform temperatures, uniform CO2 concentration, uniform growth conditions completely. And what we've seen is that the organisms grow much faster in these systems. In addition, you don't have to take the plates out to examine them. It's done with an imaging system. And for many laboratories with manual systems, they'll go in the morning, take all the plates out of the incubator, put them at the desk, They're not in an incubator anymore. They're not in CO2. And they'll sit there for hours as you work through all those specimens. In this case, they truly stay in the incubator and continue to grow, which is part of the reason why you see earlier growth. Now, reading with one of these systems allow a number of advantages. Some I've already touched on. They're in a closed incubator, allowing for more rapid growth and earlier detection of colonies. Multiple plate images from the culture can be examined, as illustrated in the picture on the left-hand side. You can also enlarge the plate that you're interested in, so you can make sure you have well-isolated colonies, and also look at morphologic features of the organism. BD has a history of working with imaging, primarily in our women's health side of the business with cytology. The same engineers that are doing the imaging work with Keystra were the ones that developed the first automated cytology imaging system. They've also developed what we refer to as Optus software for imaging. I'm going to describe that in a couple of minutes. And the imaging allows you to do what I think is important quality control. We know that in our laboratories we have some technologists that are very content with subculturing organisms and not working them up until the next day, even if there's adequate growth. Well, you actually have a permanent record of those images that you can review. You can also look at the images and use those in medical records, and I've seen some of the Keystra sites actually show pictures in the medical records of the growth in the plate. And finally, you can save images, particularly of unusual organisms, to share that with other technologists. So it's a useful training exercise. One of the studies that we've done looking at the imaging is we really posed the question, and this was with urine specimens as part of a large urine study we were doing, and we had inoculated a number of plates of media, and we asked the question, when is the earliest time that you can detect growth so you can isolate colonies, you can take isolated colonies and do your identification and susceptibility testing. So we did a series of timed readings while the plates were in the incubator. And the figure on the left-hand side shows you the results that we had for tryptic soy agar. The blue line is all organisms that were isolated, and the orange line is all of the uropathogens. The scale along the bottom is incubation time in hours, and the scale along the left axis is the percent of organisms that could be, percent of specimens that could be worked up. At 8 hours, we had greater than 95% of the uropathogens were able to be worked up for your identification and susceptibility tests. By 12 hours, all of the organisms were available to be worked up. If you looked at McConkey agar, you see the exact same result. By 8 to 10 hours, all of your cultures could be processed. So what that's telling us is that rather than waiting till the next day, you can start doing work much earlier. And in fact, many laboratories that use this are able to take this, they get their identification within the hour with MALDI, and within 10 to 12 hours, you have the majority of your antimicrobial susceptibility results. So all of a sudden, now you've completed your work with a urine culture in less than a day. And now we start to be truly innovative and valuable to our clinical counterparts. If we look at a couple of other types of media, the selective media is a little bit, or some of the selective media takes a little bit longer. CNA at about, at 8 hours we had 95% of the results. We had to extend the incubation if you're using CNA for detection of organisms. The chromogenic media, because you're waiting for colors to develop, also takes longer. About 85% of the uropathogens could be detected at 8 hours on the chromogenic media, about 16 hours for 100%. But what again it demonstrates is that you can start to get much earlier time to results with these types of systems. One of the other things that we looked at is how can you improve imaging? And this is where I get excited because this stuff is really innovative. Well, it's innovative for a microbiology laboratory. It's not innovative for what we do in life. The top figure just shows an individual looking at a butterfly, and your mind processes what it sees. And that's the point that I wanted to make with the first figure. Our mind will look around the room, and we see different things. We see some of these pictures that are black and white. You sort of have to pull out the contrast to be able to see the images on these pictures. We do that in our minds. If you have a camera and you take a picture, you get a nice image, but you can use software like Photoshop or a variety of other programs to improve the contrast, the focus, the lighting conditions with that. If you think about in your laboratory the way you would look at an agar plate with a growth of organisms, you'll hold the plate and you'll look at it when it's flat, and then you look at it at an angle where you see reflected light, and then you look at it in transmitted light to see is it hemolytic or not. So you go through a series of processes, and again your mind is processing all that. You assimilate that into what you think that looks like a group B strep, or that looks like a group A strep, that looks like an E. coli, that looks like a Pseudomonas. What we do with software is take those images and reduce them down to their pixels and do an analysis of that. We actually do it in a more sophisticated way. What we do is we take 22 images under different lighting conditions, transmitted light, reflected light, flat light, under different backgrounds, white background, black background, and that's taken very quickly. It just takes literally seconds to be able to generate that data. The software then, because we reduced all this to numerical value, can then take for each pixel for those photographs and optimize it for signal to noise ratio. And what you come up with is an idealized picture that shows you all of the things you are doing manually by transmitted light and reflected light and so forth. And so every image that's created with the Opta software and the Keystra Rita incubator is the first all going to look identical. From if you took the same plate and you photographed it in 100 Keystra incubators, all of them will be the same. Before we introduced the software, every image looked a little bit different. And so by making reproducible images that are idealized, it allows you to gain a great deal of experience in how to interpret those plates. I just want to illustrate one example, and it may not show up too well. This is a plate that was just photographed. If you took your camera and photographed the plate, this is what it looks like. And there's a number of bacteria colonies that are growing there. And so we take this area and we'll separate that area. Now, if we apply the Opta software to this plate, that's what it looks like. And so what you've done is you've idealized the actual image that exists there. This is sort of a dramatic picture. I had a whole series of other ones, but I took them out just because of time. But what you end up seeing is really very different imaging than what you've ever seen before with this. I should also point out, this is done with a camera that's less sophisticated than what's in your phones today. Now, we're going from a 5 megapixel camera to a 26 megapixel camera that'll be introduced this year, not to improve the imaging. It will do that, but actually these cameras will be much faster and allow us to generate images much faster than what we can even do today. So that improves throughput through these types of incubators, and that's the main value there. I'm going to also talk a little bit about then some of the other opportunities with imaging. You can look at a plate and say there's no growth in the plate. And the system will automatically take that plate and discard it. So you're not wasting your time looking at negative cultures, you're actually using your time to do productive things. You do semi-quantitation, I've already described how that can be done. You can do presumptive identification on chromogenic media because they can recognize colors that are present on the plate. You can measure the size of zones of inhibition around a disc. You can pick a colony, and then the system will use the data that it gathers from that colony to pick other colonies that are identical to it, so that when you do your identification or your susceptibility test, you come up with, you're picking the same organism and not organisms that may turn out to be a mixture. And then finally, you can monitor growth, as I sort of demonstrated, to say that this is the right time to select this plate so you can pick colonies and do identification. So that's with imaging. We also have the ability to use software to really monitor the amount of work that's going through a laboratory. Something I always tried to do manually and never was successful. The figure I have here is just a graph showing a series of light green bars, and that actually represents throughout the day when work was available to be done. The dark green line represents when technologists were available to do the work. And then early in the day, you can see there was work that was available, but it was not being done. And at the end of the day, you see there are technologists that really weren't working because there wasn't enough work for them. You start to be able to match the amount of work that's needed when the types of specimens in your incubator, when those specimens are going to be available for work, and you can estimate the number of technologists that would be needed to do that work. So it allows you to run a laboratory more like an appropriate operation, a business, than what we've been able to do historically. And so what I've talked about are all the things that currently exist with the Keystra system the way it is today. But obviously we also start to look for other opportunities. And so one of them is to be able to take the plates that you're looking at, and for you to select a colony that you want to have identified and do susceptibility tests, and then have a platform that automatically does that work. Because it really doesn't require a lot of thinking to pick colonies, make a suspension of organisms, inoculate a multiplate, inoculate a susceptibility panel. That should be done in an automated way. It also allows you to do this a little bit better. I said one of the downsides of one of the reasons you won't get an organism identified with MALDIs, you don't have enough organisms. What this actually does is that it will look at the number of organisms that it has and say, all right, apply the inoculum to the MALDI target plate, let it dry, apply another inoculum, and it can layer the inocula so that you end up having an adequate number so you get more reproducible profiles and you get a higher percent of identifications with this type of system. Also, because it standardizes the inoculum, that's more accurate than what we can do manually, so your susceptibility results are also going to be more reproducible. This system will also allow you to take the inoculum and use that to do disc diffusion susceptibility tests, if that's what your laboratory does, and do that in a completely automated system. Or to take that cuvette with your inoculum, and if you use a Vitek 2 card, you can take the inoculum and inoculate that directly into the Vitek card. So what these systems start to do is give you a great deal of variability, an opportunity to do different types of microbiology. And then the last thing just to comment on is you can have relatively sophisticated informatics that are applied to these systems to not only link the different components together with the different components in the laboratory, but also facilitate more rapid reporting to the physicians of data that's being generated. So that gives a very broad overview of what I look at as innovation with lab automation. So as I want you to think about automation, is really to focus on, first of all, this is going to be the future, much like you look in chemistry and you look in other labs, you see automated platforms. Microbiology will look like this. So what we want to see is that it'll help us do things that we've never been able to do before or do well before. And I think that's the true advantage of automation. So again, with that, thank you very much. If we want, we can handle maybe a couple of questions or what?
Time for about 10 minutes of questions, so 10 minutes questions.
Okay.
Basically, this is quite amazing how this automation is leading our field. But this also will lead us to